Hello everyone, it is with great pleasure that today I will share with you some information about mitochondrial DNA and how that could be used to learn more about our family history and our ancestry. My name is Ugo Perego. I have a PhD in genetics, and genetic genealogy has been my thing for the last 20 years. And of course, what I'm going to share with you in this presentation is the result of my own work, and so I'm the only one that has to be blamed for it. So mitochondrial DNA is one of the genetic markers used in genetic genealogy. The other markers, which will not be the focus of my lecture today, and other speakers will talk about that here at RootsTech, is the Y chromosome, which follows a paternal line from father to son. Only males have the Y chromosome, so if a lady would like to use Y chromosome for her family history, she need to have a male relative tested in her stead. The other genetic marker which we often hear about is autosomal DNA, which is probably the most common DNA test that people are buying nowadays. And then we have mitochondrial DNA, which is going to be the focus of my presentation. I'm going to share insights from this particular marker. This marker is inherited along our maternal line from mother to children. So the difference between mitochondrial DNA and Y chromosome is not only the side of the family tree where we are inheriting this information, but also that while the Y chromosome is only found in males, mitochondrial DNA is inherited both in males and females. Only females, though, pass it on to the next generation. So in my case, I have my mother mitochondrial DNA as well as my father Y chromosome, but none of my children has or will have my mitochondrial DNA. They all receive it from their mother. Mitochondrial DNA is helpful in family history because we know exactly where it comes from. That's the story of our mother line. So it's our straight, direct, unbroken maternal lineage that we're talking about here. So just one ancestor out of the many ancestors that we have in our family tree. When we look at the different mitochondrial DNAs found in the world population, so every single person that lives and that ever lived here on the earth have their own mitochondrial DNA. This mitochondrial DNA can be grouped in different family branches, in different, we call them haplogroup branches. And these haplogroups, these branches can be organized in some sort of pedigree chart. It looks very much and similar to what would be a person pedigree chart with the different branches, ancestors, and descendants. But in this case, we're talking about branches that have hundreds and thousands of years in age. And all the different lines, the different mitochondrial DNA found in the world today can all be traced back to a single woman who scientists refer to as the mitochondrial Eve. This is not to be confused with the biblical Eve that we're talking about. It's not the same person. This is the one person that had daughters to pass on her mitochondrial DNA at every generation for thousands of years, all the way to the present time. And as each mitochondrial DNA at each generation is passed on, then occasionally we would have little tiny changes called mutations that would create different branches and separation from one branch to the other to create what you see here, this beautiful worldwide tree representing all the mitochondrial lineages of the planet. In this little tiny square, and I know you don't see, there are not great details right now, it's very small, but we're gonna see this later at a better resolution. That is my line, it's called K1, so I belong to this particular branch, and these are all my distant relatives separated from me by thousands of years. Based on this knowledge and the development of this mutation, scientists have been able to trace the migration of the different populations and the different groups throughout the history of humankind. And so as we follow when and where this mutation took place, we're able to distinguish the path that the different groups of people follow through the years, all the way into the American continent, which is the last of all the continents to be colonized by modern humans. So this is my personal, my own mitochondrial DNA. Is the list of all the mutation, all the values that make up my mitochondrial genetic signature. And there are usually a low resolution and a higher resolution type of test that people can buy or could buy with regard to mitochondrial DNA. So when we find matches or we compare our mitochondrial DNA to that of others, what we usually are faced with is either a lower resolution or fewer markers called the hypervariable region or control region, which is the one that you see here to your left, and is divided by an area called HVR1, which is found toward the end of the mitochondrial DNA genome, and then HVR2, which is actually found at the beginning of the mitochondrial DNA genome. The mitochondrial DNA genome is only about 17,000 bases long, which is much smaller compared to our total genome, the nuclear genome, which has over 3.2 billion bases. So it's a small genome that we carry in most of our cells. Here we have the low resolution to the left, just a control or hypervariable region. And then to the right, you have the full set of values that make up my own mitochondrial DNA. So that includes the hypervariable region, which is the one that is shaded, plus other values that are found in the region called coding region. So hypervariable region, coding region, the two together is the highest level of mitochondrial DNA resolution that we can obtain. And it's very affordable nowadays. When I did mine in the laboratory, I actually did my own mitochondrial DNA. That at that time was quite expensive. Nowadays it's less than $200. You can have all this information about yourself. So what is going to be, what do these values mean? So as you can see to the left, there is a line called your sequence, which is my own set of mutation. And then there is another line, a second line called reference. So the way that mitochondrial DNA values are reported is that there is an industry standard called a reference, or call it the CRS, Cambridge Reference Sequence. You can see that in the table on your right. There is a column called CRS. And every single person in the world has their own mitochondrial DNA compared to this industry standard. And only the differences from this standard, from this reference, are reported back to you. So wherever you and the standard have the same value, that value is not reported. You can infer it that is the same. You can look it up online. You can actually see the whole reference listed, and you can look at that, but that will be information that you don't need to know. What you need to know is where you are different from that reference. And then if other people have the same differences, that's where you're going to find some matches. So it's a little bit technical, but don't worry about it, because that is not stuff that really matters to you. What it matters to you is to know that if you only have a low resolution type of test for mitochondrial DNA, then you're really missing out on some information about your own maternal line. So what we have here is a snapshot of a large tree that organize all the different branches of mitochondrial DNA. And I told you I belong to a branch called K1. And because of a particular mutation that is found in the low resolution level tested, the control region level, which is called 497, because I have that mutation, then I can go as far as saying that I am in a branch called K1A. But if I have a full sequence, then with the full sequence, I can get 3 extra level of depth and narrow down a little more the group of people that share the same mitochondrial DNA with me. So I'm actually a K1A4A1. So the 4A1 comes because I was able to have the high resolution test, and so to define and refine better the type of mitochondrial DNA that I belong to. With that information, I can find more specific matches and not some false positive or general matches. The way it works is very much like a family tree. Whenever we have a mutation, so the absence or presence of a mutation, create a very specific journey along that tree. So we start with, I was saying, I'm part of K1, and then the presence of a particular mutation puts me down one extra level, K1. And then what I have some sister clades, some sister lineages called K1C, K1B, and we are all daughter of a parent lineage called K1. And on the next step is the same thing. So you see there is a sort of hierarchy based on the absence or presence of this mutation, all the way to the most specific lineage or subgroup where we can put. So that is mine, K1A4A, and then anybody in the world will be in one of these different branches. So that provides some information about our ethnic origin. This is called a heat map. People that have been tested for their own mitochondrial DNA all over the world. So if I want to know where the people that belong to my same mitochondrial DNA line live, then scientists have tested many different areas of the world. You can see there are none in South Africa, there are none in Asia, there are none in the Americas, there are none in Australia or Australasia, where they're mostly found in Europe. And then where it's darker, the color, that is where you find the most of these people. This is a map that comes from 23andMe, which provides you some sort of survey general information about your haplogroup. It will tell you the ethnicity value of your mitochondrial DNA, but it will not tell you the individual values and the matches of with other people at a more specific level. That's only another company does that. But this is an interesting map that I found very helpful to give a glimpse about the distribution of my particular mitochondrial line. The only company that provides mitochondrial DNA testing is Family Tree DNA. And again, is very affordable. I don't work for Family Tree DNA, but that's the only company that provides this type of test. So there is no other choices out there. They provide a full sequence with all the individual values. And so, in fact, this is my own account. This is my own test. I tested my own mitochondrial DNA in the lab. I did my whole the full sequence like you saw in the table before. But then I also wanted my Family Tree DNA to do it. For one reason, I could double check that I did the work I did was correct. But they also provide me a way to compare my mitochondrial DNA with that of all their customers in their own database, and so I can find possible matches. So I'll show you some of the things that you can see when you're tested with your mitochondrial DNA, Family Tree DNA. So there are several type of icons that you can click. Some are more helpful than others. The two I'm focusing on that I find more helpful is the set of mutation, which is right here. So this is the set of mutation that makes up my mitochondrial DNA. All these values together create my mitochondrial DNA profile, or in a scientific jargon is called the mitochondrial DNA haplotype. But profile or fingerprint will be just probably as good in genealogy. So you see here the control region, the low resolution. You could find people that only have had this type of test in the past. Many of the customers in Family Tree DNA in the past only bought this type of test. Or you can also have the coding region, which is what I purchased from them. And all these mutations match the mutation that I did in the lab, the one that you saw before. So that's something the Family Tree DNA will show you. But how does that apply to my genealogy? Well, we need to go to the other option, back to the dashboard, the home page, and click at matches. And so what we see here at this level now, we can have the complete mitochondrial DNA sequence, my full profile, and look if there is anyone else that also tested with their own full profile in their database and see if we match. And I have only one individual that I'm a close match, and in the first column it tells you the genetic distance, which is how many differences between my mitochondrial DNA, or the total amount of values, and this person mitochondrial DNA. So the two of us have 3 differences. Considering the very slow mutation rate of mitochondrial DNA, which is in the range of 1 mutation every even thousands of years, having 3 differences from another individual means that we are related on the maternal line way back in time, before genealogy and genealogical data and records were kept. So yes, I share a maternal line with this individual, but probably too far back in time to find a way to prove that connection. And so it's okay. She's also is a or he, this, you know, that this maternal line is the same I have, is a K1, A4, and 1, and then they have their most distant common maternal ancestor, which doesn't resonate with me. It's not anyone I know. But if I click on the name, which I cover for privacy purposes, then I will see the email address for this person, so I can write to this person, and I can see also the origin of their ancestors, which are mostly from Portugal, and I am from Italy, and so I don't recognize the name, the countries are different, and the 3 differences between my mitochondrial DNA and this person's mitochondrial DNA probably means that we are not going to find a connection. Now, let me open a parenthesis really quick. Mitochondrial DNA testing is often more helpful to disprove relationships that we think are in existence than to prove relationships. So the fact that 2 people have an identical mitochondrial DNA sequence does not mean that they are closely related. They are related, but they could be related very close in time or very far back in time. And so by 2 people you think are related have different mitochondrial DNA, then you know they are not related. And that is going to be a proof to you that you can use genealogically to exclude that relationship. There is an option where I can actually remove the coding region and only look for individuals that have my the low resolution type of test, the control region. So you see here there is a drop down window on the top part of the menu. Let me get the laser right here. HVR1, HVR2, so I remove the coding region from the equation. So now I have 36 matches right here. 36 matches. I'm going to show just the first few. These have to be identical matches. There are no longer 1 or 2 or 3 differences. Only identical matches are listed. And as you can see here, there are many people from different places, maternal ancestors from different places. Again, what I have to do is to contact these people if I want to find out something more about my genealogical connection to them. But listen to this, you know, mitochondrial DNA without a genealogical context, an historical context, something you want to test, just looking blindly in the dark for possible connections, mitochondrial DNA is not going to probably do it for you. But if you have a situation where you need to verify some genealogical historical information, then mitochondrial DNA can help you. And I'm going to show you a couple examples in conclusion of this presentation. The first one is the case of King Richard III. You probably heard about him. His remains were discovered in a parking lot, and during some excavation, totally random, they were found and no one knew where King Richard was actually buried. There was no official burial spot for him. They found his skeleton. The skeletal anatomy matched some of the historical records we have for King Richard. Remember, he lived in the 15th century. So we're talking about 500 years and more ago that this person disappeared, this king disappeared. And there are wounds, how he died, and that also match the historical records. But in order to verify if the skeleton really belonged to King Richard, they used some genealogical data And so what they had, King Richard, even if he had children, they would not carry his mitochondrial DNA. But what we have is a sister, Anne of York, of King Richard, and she has descendants, including direct female lines, all the way to the present day. And so two individuals were found. They were separated by several generations, 13, 15 generations, removed from Anne of York. And they tested both individuals, and they both have the same mitochondrial DNA haplogroup, which also was the haplogroup that King Richard had extracted from his bones. So they tested the bones and they had this haplogroup. And then they tested two descendants of Anne of York, same haplogroup. So we have now a beautiful pedigree chart. We have some evidence from the anatomy of this individual, and also we have the mitochondrial DNA, which supports the genealogical finding, the historical finding. There is a case I had a chance to work on a few years ago. It has to do with a very sad historical event that took place in southern Utah, known as the Mountain Meadow Massacre. It involved it took place in 1857. We're talking about far west America, cowboys times, the colonizing of the West. And there was a group of people that was moving from Arkansas to California, and they met their fate in southern Utah when they were attacked. 100 people were killed, and a small number of children survived, were spared, and were given to local family to raise as their own. And among these, there was a girl that eventually was never found. There was rumors of a girl that was kept by this family belonging to Philip Klingsmith, who was one of the persons that was associated with this massacre. And she was identified as Priscilla Klingsmith. And she was born in 1855. No birth records were found about her. Again, we're talking about the 1855 Utah. I mean, there is not even Utah existing anymore, so records were probably not kept properly at that time. But there is this girl, and then rumors say, well, she's one of the children that survived and that was never returned to her relatives. And so what we did here was to identify some biological relatives through the maternal line. And so we did a mitochondrial DNA test on a direct descendant of Priscilla. And then on her, Priscilla had a younger sister that was born in 1859, so 2 years after this massacre, so no association between the birth of this girl and the massacre. And we tested a descendant of that, and then as a control, we tested also a sister of the mother, the putative mother, Betsy Cato. So we had 3 individuals that apparently genealogically are all related, and they should all belong to the same maternal or mitochondrial line. And here we have it. We have the mitochondrial DNA of the 3 individuals. It's a low resolution test, but it's a very specific mutation that these individuals share. And considering the low population at that time, and the fact that they all belong to the same family, and the probability that this happened just by chance being very low, then we can conclude that this girl was actually the biological daughter of the family that raised her, and that was not one of the surviving children of that massacre. So there are different things that come lesson. I hope you understand the mitochondrial DNA has some benefit genealogically, both in telling you something about the ethnicity or the geographic origin of your line, as well as possibly be used within a genealogical context. Also, there is a way to bypass the fact that we only receive that from our mothers. For example, in my pedigree chart, I have 3 different mitochondrial line represented, and that's because I had my paternal grandmother and my paternal grandfather also tested, and they belong to different mitochondrial line than I do, but they also this mitochondrial line would also be part of my family tree. I just don't carry that DNA, but it is my family. So you see, every line belongs to a different haplogroup, which means I have a much richer ancestry with regard of ethnic origins and genetic cousins that I can find along these different lines. So that's all for mitochondrial DNA. I hope you enjoy it, and thank you for listening to it.